Good afternoon, everyone. Welcome to today's seminar. It is my pleasure to introduce today's speaker, Professor Vahid Sandoghdar. Vahid is the Alexander von Humboldt Professor of Physics at the Friedrich Alexander University. He's also the director of the Max Planck Institute for the Science of Light, as well as the founder of the Max Planck Center for Physics and Medicine. Professor Sandoghdar is one of the pioneers of the field of nano-optics, which merges different methods and different research areas to study the fundamentals of light-matter interactions. Currently, Vahid's research focuses on the areas of quantum optics, nanobiphotonics, plasmonics, and ultra-high resolution microscopy. Before I turn it over to Vahid, I just want to remind our audience, you can keep your questions to the end of the seminar. At the end of the talk, you can ask your questions either by typing them through the Q&A feature of this webinar, or you can raise your hand and directly ask your questions, and you'll be able, and we'll be moderating the Q&A session. So with that, Vahid, thank you very much for being here today. We're very excited to hear about your great research. I'll turn it over to you.
Thank you very much, Farnaz, for the introduction, and most importantly for the invitation, the opportunity to speak to everyone at MIT Nano. As Farnaz said, our work kind of covers many of the things that are done under the umbrella of MIT Nano, and that's why I'm particularly excited to talk to you guys today. So I've just tried to bridge the different things that we do, and that makes the talk maybe a little too general and less technical. But if anyone has questions, then I'll be happy to go into the technical aspects afterwards, or you just get in touch with me offline. So let me start by kind of telling you where we are in Germany. I think you know Germany within Europe, but Erlangen is a small university town. It's not a touristy spot, so you might not know where it is. That's Frankfurt and that's Munich. That's about 2-hour distance from each one. Erlangen is actually the seat for many very global companies like Siemens Healthcare and Adidas and Puma and many others. Again, it is a university town. The most important feature of Erlangen is probably our institute, which was established in 2009. This is the new building that we moved to in 2016. And we are excited about expanding our efforts in the direction of the interface between fundamental physics and fundamental medical research. And we have just started building a new building dedicated to that on the site of the university hospital. So towards the end, maybe you get a little bit of flavor of why I'm myself interested in this, but there would be great opportunities there to bring in maths and physics together to actual medical research. Okay, so with that geographic introduction, I want to thank the people who are working with me right now before I forget that at the end. Our group has kind of two sections. One concentrates more on nano-quantum optics, and these are the people who are involved. The first row are staff colleagues, and then we have postdocs, the second row, and PhD students, and the third row. And we also have by now a sizable effort in biophysics, and again, some permanent staff contribute to that, and the second row are postdocs, and then a good number of PhD students. Hopefully by the end of my talk, you will see why and how we've kind of gotten involved in biophysics or let's say my background was originally more from atomic physics and quantum optics. But this is now a very exciting area of activity for us. Okay, so let me start my general talk by, let's say, having a look at the issue of signal processing and of course, electronics, microelectronics has played a very important role in that. When you have an electronic circuit, then you have lots of different components like capacitors and transistors and resistors and switches and all of that, that actually come together to make it possible to process signals and information. Now, this has over the years gone through a huge revolution in terms of fabrication and miniaturization, and of course, electronics has become more and more powerful now. But as an optical physicist, you're interested to do this using photons instead of electrons, and this has been actually a goal since the 60s, and its progress has been much slower than the progress that you've seen in microelectronics. And here's a little bit the reason for it. The optical elements are usually quite bulky, and even when you get to micro-optics, you're still talking about very, very large things of dimensions of several hundreds of microns. So here, for example, you would have a small laser. You could make it much smaller than that, but still huge. And so one important goal is to miniaturize all these things in optics and integrated optics. But one of the things that I will be, or we've been emphasizing, is not so much miniaturizing these elements, but realizing that even if you miniaturize all these things, one mundane but very important feature of any circuit is done by basically wires. And wiring optical elements is a very challenging task because, of course, you don't want to lose your signal. When we get to light-matter interaction at the very small scale, if you were to miniaturize all these things at the nanometer scale, then you would eventually get down to single photons and single atoms or single molecules, and that's basically where we want to go. And as you will see in the next few minutes, the efficiency of interaction between these two becomes a huge issue. Now, this in the more of a kind of a modern language becomes very important, and you will see that also the way the kind of accent in our own work emphasizes that this interaction between single photons and single emitters is obviously central to the emerging quantum technology, but it's also very important in biotechnology or biophotonics, where you would like to detect and sense individual biomolecules, in our case, using light. All right, so let's get down to it. If you want to really understand light-matter interaction at the fundamental level, then you would have a photon and you would have a two-level atom. That would be the simplest type of matter. Typically, you write a Hamiltonian that consists of the electric field of light at the position of the atom and the dipole transition for this two-level system. And you go on. Now, this is what is typically the approach that is typically done in quantum optics. There is an equivalent way and slightly more maybe intuitive picture or phenomenological perspective, where you would say that, okay, let's just think of my particle. This could be a single atom or a nanoparticle, and now I'm illuminating it. And basically, I want to know what happens during the interaction. Well, the most fundamental interaction is scattering, but it could be that your system also dissipates. And if it dissipates, then that light is removed from the incident beam, and then you would say that you have absorption. Either way, the interaction can be described using a, let me get my laser pointer here. The interaction is described using a cross-section, sigma. And the amount of power that is attributed to the interaction would be the incident power, P0, times sigma over A, where A is the surface area of the light that you've used. So this is the simplest kind of light matter interaction scenario. You would have a detector in transmission, and you would know, you would learn something about the interaction of light and that particle. Now, in the case of a classical particle, this is the textbook physics that you would know. The amount of light that is scattered is proportional to the incident light, with the polarizability being the proportionality factor. The polarizability is proportional to the particle volume. That kind of makes sense. If the particle is very small, the polarizability is small, and vice versa. It, of course, also depends on the dielectric function of the particle and the dielectric function of the medium. But this contrast here, this fraction here, is not going to make a huge difference. So the real thing is the volume here. Now, there's a scattering cross-section that you attribute to this particle, and there's an absorption cross-section. And either way, you see that alpha, your polarizability, is the central parameter of interest. Now, if you go to a right, and it's important here from a fundamental point of view to realize that all the information is actually scattered in this scattered light. That is, even if you're talking about absorption, the information that is encoded on the detector comes from the scattered part of light. So what happens is that you have, let's say, something like a plane wave as incident beam, and the light that is radiated typically in form of dipole radiation, because your particle is a Rayleigh particle when you talk about nanoparticle, or you have a dipolar transition. Either way, on your detector in far field, you would have intensities associated with each one of these, but since this is a coherent process, you would also have a cross term, and that would be the so-called extinction term in scattering theory. Now, the cross-section that is attributed to a single two-level system is actually remarkably large. It's given by 3 lambda squared divided by 2 pi, so roughly lambda squared divided by 2. This is something that you can look up in your standard textbook on quantum optics. You can also find it in Jackson. So this cross-section from a single atom is actually very large, but you have to realize that in this case, there is no absorption, because all the light that is scattered, that it interacts with the atom, is scattered. Now, if you work out the details, and I'm not going through all the details here, but if you work out the details, then you can show that, in fact, if you have a single oscillating dipole, and you put your light not in plane wave, but in a mode that is the same as the mode of a radiating dipole, and use that as incident light, you can get 100% extinction in the forward direction, which means that all of the light is reflected backwards. So a single dipole acts like a perfect mirror. This is to say that and it's the central point of my talk, is that the intrinsic efficiency of interaction between a photon and a single atom is extremely high. Now, in practice, you will see that this doesn't happen so easily, and we'll talk about the reasons for that. One reason is that you usually have a hard time putting all your light in a dipolar mode, but if you were to focus within a very high NA, you would put something of the order of 85% of your light in the dipolar All right, now one reason that this doesn't happen on a daily basis is that there is another fraction or factor in front of this cross-section term that I told you about, and that has to do with the ratio of the radiative loss to the overall line width. So typically, if you have a two-level atom, gamma 0 would be the spontaneous emission rate, and gamma would be the sum of all the dephasing or decay processes. If you put your emitter in a matrix at room temperature, because of the thermal jitter that you have in your matrix and the phonons that are interacting with your emitter, you get a huge dephasing effect. And therefore, typically, this ratio that we're talking about is of the order of 10 to minus 5. So that's why it's not easy to see scattering from individual molecules at room temperature. Now I'm going to show you how we actually get to a point where we interact very strongly with a single molecule. Everything that I've told you so far was quite general in terms of the matter side, that is whether you have a quantum dot or an atom or molecule or a color center, the physics is the same. Now in our lab, we like to work with organic molecules quite often, and organic molecules were detected at the single molecule level already almost 30 years ago. And this is the thing that you probably mostly know from what you've read or talks that you've listened to. You can, if your molecules are spread around, then you can use fluorescence to detect individual molecules. So each molecule gives out a small amount of light, but if you manage your background and have good detectors, this signal is sufficient for you to be able to detect single molecules. Now, the typical molecules that you might confront, especially if you're doing biophysics, are from this kind of a family of molecules, and it's well known that these emitters are not particularly good in terms of photostability. They bleach and blink, so they might have a kind of a bad reputation in terms of a quantum emitter. Now, what I want to kind of impress you with is that there is another class of molecules that actually was detected before the typical type of molecule through the work of W. Myrna and Michel Horry about 30 years ago. And that type of molecule is a dye molecule, but it's a little bit like having cut about in a graphene sheet and then passivating it with hydrogen. So these are rigid planar molecules, and they turn out to be extremely photostable. We can work with one single molecule for a couple of years if you want to. Now, the typical level scheme is just like any other molecule. You have a ground state and an excited state. And of course, you have vibrational levels associated with that, which allows a fluorescence channel. Now, when you put these molecules in a crystal or in a matrix, the vibrational levels become very lossy. So they decay within a picosecond or so. That means that this channel here becomes a lossy channel, or you can say that there is a lot of decoherence associated with it. Nevertheless, if you go to low temperatures, to temperatures below 2 Kelvin or so, the transition here that is shown by yellow becomes extremely narrow. It becomes so narrow that, let me run this again, it becomes so narrow that the individual lines of the different molecules in the samples no longer overlap with each other. That means that if you scan your laser frequency through, you will see how different molecules light up at different places. Okay, so the the pointer down here is scanning the frequency over something like 50 gigahertz, and you see how molecules at different places light up. And it's in this combination of this type of molecule and an organic crystal that makes the system extremely photostable at cryogenic temperatures. And in addition to that, at this cryogenic temperature, because the lines are extremely narrow, that means that all the coupling to the matrix has been essentially frozen out, and we can reach a cross-section that is close to that fundamental cross-section of lambda squared divided by 2 that I told you about. So this is the cross-section that we have, and now you can see that if you could confine your light to the same type of area, you would expect to have essentially a large shadow from a single molecule. That is, you would have a very efficient interaction between an incoming beam and a single molecule. Now, confining light down to something of the order of lambda over 2 is not that amazing. It's basically within the diffraction limit. Except that if you want to do that in the cryostat, it's a little difficult. At room temperature, this is the typical microscope objective that you use to get a very high numerical aperture. If you can't put that microscope in a cryostat, we had to devise our own microscope objective, which is actually quite simple. Goes back to one of the first demonstrations, also 30 years ago. You use a half-ball lens as an effective solid immersion lens, and then you can use a cheap aspheric lens to couple light in. And now, of course, we have to prepare our sample on the flat side of this solid immersion lens. We've done that, and this is a series of works that we've published over the past 15 years or so. Here's the result in a nutshell, the basic result in a nutshell. You see that a single molecule is extinguishing an incident beam, okay, in transmission by 10–12%. Okay, so what is happening here, you see here also how narrow a single molecule line can be. It's of the order of, in this case, 17 megahertz. You detect a single molecule, although there are lots of them in frequency space. You scan your frequency and you see that when you're resonant with it, you get a large amount of shadow. Again, once you are in this regime, you're doing coherent scattering or Rayleigh scattering very efficiently from a single molecule. And that coherence that you have helps you do a number of things like shifting the phase of a laser beam, or doing very efficient nonlinear spectroscopy, because now each photon is interacting with your molecule in a very efficient way. A molecule is not a two-level system, though. We just discussed that briefly. And the important thing is to remember that in addition to the main transition that gives you that effective Rayleigh scattering, coherent scattering, there is this dephased or decohered channel that comes via the vibrational levels of the ground state. And that can be summarized in something like a branching ratio that we call alpha. And it turns out that this alpha is of the order of 30% or 50%. That is, only about 30% or 50% of light comes down via the main transition. The rest goes via fluorescence. This is just a property of the molecule, the way things like Franck-Condon factor work out. And there isn't much that you can do with it. If you look at any other type of emitter, you see that you have some kind of an equivalent situation in almost all cases. So having a two-level system is actually not very easy to have in the lab. Now, this is the situation that I just described, and now the question is, how do we deal with it? Well, we really want to have almost 100% interaction efficiency, because remember that one of the motivations is to do things like signal processing, and you want to do that with at the level of single photons and single quantum emitters. And there you really can't afford to have large losses. So the way to do this is to enhance this transition so that the probability of decaying via this transition would become a lot larger than usual, and therefore compared to that, transition via fluorescence becomes negligible. And the way to do that is well known in cavity quantum electrodynamics or quantum optics. You basically just want to couple your molecule to the mode of something like a microcavity. Typically, the microcavity physics is described in the language of James Cummings' model. But again, I can use my cross-section language in a pictorial way, and it's actually quite accurate. So what happens is that you come with a photon, the photon goes into the cavity, and now it can circulate many, many times, given by the finesse factor F. And now every time the photon goes by the atom, it has a certain probability of interacting with it, which is given by sigma over A, where A is now the beam waste inside my cavity. So at the end of the day, the figure of merit is the finesse divided by the beam waste area. Typically in a microcavity, you win because your F can be quite large, but you also lose in a typical microcavity experiment because your omega squared is much larger than the diffraction limited spot. Anyhow, when you, if you were to reformulate this, you would show that this is the same as Q over V, where Q is the quality factor of the microcavity and V is its mode volume. And that, of course, is what you would know as the so-called Purcell factor that determines how much spontaneous emission is enhanced. You can also rewrite that and show that this is the same thing as the so-called cooperativity written in terms of G, which is the coupling rate between the photon and the atom, and kappa is the loss rate of the cavity, and gamma is the loss rate of the atom. Okay, so that's kind of the framework. Now what we want to do is we want to put our molecule in a microcavity like that. And it would be sufficient if we could enhance this transition by a factor of 10 to 100, because we are already at something like 50-50. And if we could do this 10 times or more, then it would be more than 90%. There are some technical issues when you are working with microcavity at something like 2 Kelvin. It is preferable not to have a Q that is too large. So we have emphasized a route that tries to minimize V and not maximize Q. This is where we want to go. We want to actually minimize V and try to keep Q somewhat moderate. Either way, you would get a large Purcell factor. And our approach is based on an open Fabry-Perot cavity. There are lots of other ways of making a microcavity. They have their own technical advantages or disadvantages. The great advantage of an open microcavity, the way we do it, is that we have full control over that in situ. So the idea is that you want to make a mirror that is extremely small but curved, so that the overall mode volume can become very small. You can bring this a highly curved mirror very close to a flat surface, flat mirror. The way we make these mirrors is using focused ion beam milling. We can do that, for example, at the end of an AFM cantilever tip or at the end of an optical fiber. Once you've made that, you send it to a company to coat this. So you have, in this case, 12 layers dielectric coating to make this highly reflective. You can also see that in this process, we managed to make these mirrors extremely smooth. So overall, the smoothness and the high reflectivity gives us a very large, sufficiently large Q. So when we put this together, and again, I'm not going through the technical details, when we do that, then we get the results that we actually want. This experiment worked almost as planned. So here I'm showing you the cavity transmission and cavity reflection signal. So this is if you had no molecule in it, there would be just the so-called empty cavity. And now if you tune the resonance of the cavity to one of the resonances of the various molecules in the sample, then this is the transmission and reflection signal that you would get. So you see here that a single molecule has managed to completely change the transmission of the cavity. Namely, it is making the transition less than 1% of what it was. So this is great. And again, I'm not going through all the details, but you can read about that in this paper. Because we have control over the cavity, we could look at the very same molecule without the cavity. And you see here that that molecule has a transition that is of the order of 40 megahertz broad. That's a typical molecule at this temperature. But that same resonance coupled to the cavity is now more than 600 megahertz broad. And this is, if you want a visual proof that there is a large Purcell factor involved. So these are some of the figures of merit that you might be interested in. So we reach a finesse of about 17,000 and a Q of about 140,000. And the signal that we get here is basically at the onset of strong coupling. We actually have managed to go into strong coupling, and that paper is not is now being drafted. One of the interesting things that happens when you do that is it regards the photon statistics. You know that a single molecule emits light that is anti-bunched, but when you couple light to a molecule that is coupled to a microcavity, you actually get to a very interesting regime of super bunching. And basically what happens is that you're sending coherent light, which has Poisson statistics. That means that it's of course weak. Sometimes you have one photon, sometimes you have no photons, sometimes you have two or three. And but the composite system of molecule and microcavity is very efficient in reflecting the single photons. And that means that the light that is going through now is typically bunched. They are not single photons, but there are many photons at the same time. So we get to a G2 that is more than 20. All right, so that is kind of in a nutshell a report on an experiment where we showed that we could turn a molecule that has a complex internal structure into a two-level system, which effectively was extinguishing all the light that was coming onto it. Now, it's important again to note that there's nothing special about the molecule. Everything that we've done is quite general. You can put any kind of quantum emitter and that would work. In particular, there are people who are interested in improving the optical properties of their color centers, or nowadays 2D materials, or quantum dots, or ions, or anything else. Now, once you do that, you can actually now try to replace your laser light with single photons to really get to a regime where you interact single photons and single quantum emitters. Well, if you have the one way to generate single photons is to actually have a single quantum emitter, in our case, a single molecule. So when you excite the molecule, it can only emit a single photon at a time, which makes it by definition a non-classical source of light. And so this is the experiment that we have realized. We have a molecule that we use as a source. We generate single photons, couple them into a fiber, go to a different lab and couple those photons to the first molecule that we had coupled to our microcavity. So again, there are two labs. One lab has a cryostat that acts as a single photon source, and there's another lab with a cryostat that had the whole microcavity establishment. And all we have to do is we have to replace the laser kind of part of the optical setup with light that is coming via fiber into the cryostat. And you end up getting very similar results, the same type of efficiency. And it's just to show that we're now at a level where we can interact single photons and single quantum emitters. Now, when you talk about single photons, of course, efficiency becomes very important. You generate, you kind of try to generate single photons, but only once in a while you catch one, and mostly because your collection efficiency is not very good. For example, it's limited by the numerical aperture of your lens that captures the photons from the single molecule. So we have shown that by devising a particular antenna, you can actually capture more than 70% of all the lights, including the detection efficiency. So that's just a kind of an en passant thing to show that there is quite a bit of engineering that is needed if you want to have very efficient single photon sources. Now, the next thing would be not to couple one molecule to another molecule, but to couple many molecules to each other via many photons to make things like quantum networks. And having different cryostats in different labs becomes very cumbersome. So you really need to do an on-chip type of circuitry. And the essence of this is demonstrated here. So imagine that you have something like a waveguide, and now you couple individual molecules very efficiently to the mode of the waveguide. And it turns out that by the same kind of cross-section argument, you can show that now each molecule is coupled very efficiently to the mode, which means that when a photon is emitted, it bounces back or scatters from all the other emitters also extremely efficiently. So in theory, you could also make this have 100% efficiency. In practice, we have the same issue as before. And I'm not going to go, I see that I'm running a little bit behind my schedule. So I'm not going to tell you about the details of this graph, but you can consult this theory paper that works out the details of light matter interaction depending on the density and disorder that you would have in this type of a one-dimensional system. Now, we need to improve light matter interaction in this type of 1D system, just as before, using resonators, because again, remember that the intrinsic kind of property of any kind of emitter is not that good. So you need to enhance it at the end. And you can do that using resonators. Now you have on-chip resonators, for example, a ring resonator, and we would have molecules that are coupling to that. And we've shown in this paper the first results where you can detect single molecules coupled to this arrangement. The efficiency is not 100% yet, and we're working to improve that. And I hope that we will be able to report something in the coming year. Now, once you've done that, you need to also tune the molecules to resonance with each other. So this is a typical kind of spectrum that we get from our one-dimensional systems right now. But this inhomogeneous distribution of resonance frequencies that I told you about in the beginning is sometimes helpful and sometimes a little bit of a bother. So if you want to couple many molecules to each other, then it would be much better for them to have a common resonance frequency. And we do this by putting microelectrodes, and that way we can tune individual molecules through Stark effect and tune them to each other. So this is also work in progress. It looks quite good, but I'm not going to show you any results today. And finally, in the last few minutes of my talk, I want to show you how what happens if we now leave this quantum optical regime that we reach at cryogenic temperatures and come to room temperature. And this is mostly the work that we do in the biophysics area. You have materials that, usual materials at room temperature that might absorb and fluoresce. So those are, for example, dye molecules. You could have materials that absorb and don't fluoresce. And you could have materials that don't absorb and don't fluoresce. This, for example, the last one is something like a piece of glass. It doesn't absorb much and it doesn't fluoresce. And but you have to remember that everything scatters because you have a combination of electrons in your matter and they respond to an oscillating electric field. So everything scatters. And this is kind of our angle of approach. So in this slide, I'm summarizing work that is now almost 10 years old, where you see that we could manage even a room temperature to detect single molecules in transmission, that is in extinction. The signal is quite low, it's at the order of 10 to minus 6. So instead of 10% absorption or extinction, we're having now 10 to the minus 6 extinction. But if you do your experiment right, then you can show that you can detect these tiny signals. Now, this was a dye molecule that actually had an absorption band, but using the same kind of idea, you can now look at material that don't fluoresce and don't absorb. And basically, when you're doing your experiment in transmission, this is what is happening. This is what I've discussed so far. You send your light, you focus it onto a dipole, and then you look in transmission that you have a certain dip. You could, in principle, do this also in an imaging scheme where you just see the shadow of the dipole. Now, what we're going to do is we're going to flip this and put the detector in a folded way, but the physics is not going to change. So now we're illuminating the dipole and detecting a certain amount of light that is scattered by the dipole and amount of light that is reflected by this substrate on the detector. You still have extinction. And I've told you about the details of polarizability. This is a method that we call interferometric detection of scattering, short ISCAT. It goes back to 2004. And the physics is the same as what I showed you with a single molecule, in the sense that the signal on the detector is the sum of the incident electric field and the scattered electric field. And again, you have a cross term, which is the extinction term. This is an arrangement that happens very often when you're doing microscopy in the lab. So you have your nanoparticle on a cover glass, and as you illuminate your sample, you get some residual reflection, which acts as your reference beam or incident beam, and then amount of light that is scattered by that nanoparticle. These two fields interfere with each other on the detector. Let me not worry about that. So this is, again, work that we published first in 2004. In that work, we were actually interested in detecting very small plasmonic structures. And soon, if you're interested in more details of ISCAT, then there is a longer chapter, book chapter in this book, and there's a mini review in Nano Letters that I wrote last year. So we realized that we can actually extend this to biological particles beyond plasmonics. But one thing that we also realized was that we were so sensitive that we were limited by a kind of a speckle noise or speckled background when we were doing that original experiment. So here you see gold nanoparticles on a cover glass, and you see that a 5 nanometer gold particle gives a signal, but the modulations of the background are quite comparable. And it turns out that this has to do with the tiny roughness and index modulation in your sample. So the challenge now in detecting smaller objects is to mask this kind of a speckle background. And the way to do that, one way to do it, is to encode the information in the arrival time of your bioparticle. Here you see our first efforts where we looked at viruses. Viruses are quite fashionable these days, unfortunately. And we're extending these works actually to the coronavirus right now. But this is very nicely showing how a virus is actually a quite large nanoparticle, in this case 45 nanometers. And although it's immersed in water, which means that the index contrast is not very large, the polarizability of this particle is large enough to give you a sizable signal. Let me do this again. So you see here the diffraction limited spot of a single virus without having to label it, and as it's moving on a synthetic membrane. So we're quite motivated by this, but at the same time we wanted to see how far we go. Can we also detect single proteins? Well, a single protein has a polarizability that is about 1,000 times smaller than the virus that I just showed you. And you can see that either in terms of volume being, the diameter being 10 times smaller, or think about the mass of the protein. Either way, we undertook a lot of effort and improved our signal-to-noise ratio and data analysis and all of that. And indeed, we could manage to see single proteins. Here, I'm showing you the final result of that. So we have a surface, and we introduce proteins that land. Right now, the valve hasn't opened. At some point, you will see on the left side, there we go, there are dark spots that appear in the video, and each dark spot is the footprint of a single protein, or a shadow of a single protein. And on the right, we're registering every time when that protein lands. You can do this for proteins of different sizes as a kind of a benchmark, and we could show that yes, indeed, the signal scales linearly, which means that you can actually use this experiment also to weigh your proteins once you calibrate your setup. Now, there's been a lot of interest, understandably, in developing biosensors, and in particular doing optical sensors. The gold standard right now is still based on surface plasmons. This is the kind of a device that you would use, that pharmaceutical industry uses. There are other approaches. For example, people have shown that the same way that we enhanced scattering of a molecule, you can enhance the scattering of something like a bioparticle. So you can do sensing by an optical microresonator, or sensing by plasmonic nanoparticles. In either case, you try to look for a change in the resonance of the system. But these approaches are actually limited intrinsically, because you always have a certain hotspot that responds to your protein. So a lot of the proteins that are sitting on this kind of a microsphere, in this case, are not giving you a signal. They have to sit exactly in that limited area of the mode for you to get it. And the same way here in the nanoparticle, if they don't sit at the hotspot of the field, you don't get the right signal. But the method that I described to you has the great advantage that wherever they sit on that big surface area of our cover glass, we get a signal from them. All right, right, and when you do the physics, then you can show that there is a very nice analogy between the classical and quantum optical type of cavity enhancement, and in both cases, your signal also, in this case, your signal is essentially proportional to Q over V. Okay, as a very last thing, I just want to show you an experiment where we are detecting proteins, this time not directly, but by putting a gold nanoparticle as label. The reason we do this now is that we're not just doing protein sensing, but we're trying to look at a protein that is an integral part of a cell membrane. And of course, the cell environment is very busy and has a lot of background that doesn't allow us to detect the protein in an unlabeled way. So we label it with a small gold nanoparticle and otherwise do ISCAT on it. And this is now an example of a trajectory of the gold particle. In each frame, we have a diffraction limited spot, like the virus video that I showed you, but we can find the center of that with great precision. So our localization precision is typically a few nanometers, and we can do that extremely fast. Oops, sorry, this should be 20 microsecond temporal resolution, not millisecond. We can do that with extremely fast temporal resolution because there is a lot of light that can be scattered, and therefore you can integrate very fast. In comparison, this is a typical kind of signal that you might get via fluorescence, for example, using a quantum dot as a label. So you clearly see that we have a much better temporal and spatial resolution, which means that you get a lot more data and you can record your light for a very long time, essentially unlimited, because your gold particle doesn't bleach. So this is a typical type of analysis that we can get out of this. Because we have a lot of data, we can do quantitative analysis on the diffusion properties of this protein. For example, find regions where diffusion is clearly sub-Poissonian. And finally, I just show you a flavor of this, how we get three-dimensional trajectories, and that has to do with the fact that we're using an interferometric method, and so motion in Z modulates the contrast of the signal in ISCAT, and that way we can chase a nanoparticle with a nanometer resolution also in the axial direction. And this is showing you the contour or the trajectory of this protein that was labeled with a gold particle. And it's showing you clearly that this protein is just not just going anywhere, but it is going around a kind of a bowl structure. And there's reason to believe that this bowl structure is actually the clathrin-coated pit in this cell, which is where this type of protein gets internalized. All right, with that, I quickly summarize. So the take-home message that I want you to remember is that the sensitivity of light-matter interaction is intrinsically very high. So light is actually extremely good sensor for matter, and vice versa. And we're now entering an era of controlled multi-emitter, multi-photon interaction, and also you can expect important advances in label-free imaging of nanoparticles in complex media. I just showed you an example of a cell, but you can take that now into tissue and all of that. And with that, I thank you very much for listening. I'm sorry if I've gone over by a few minutes.
Thank you, Vahid, for a great talk. We have a few minutes for questions. So again, if you have questions, please type them through the Q&A section of the webinar. You can also raise your hand and directly ask your questions. So I'm going to start by some of the questions that have been written in the Q&A session. So one of the questions from Professor Jablanovic from UC Berkeley says, for the metallic structure, it's not obvious how to define the effective volume in the Purcell factor. How do you proceed in that case?
That's a very good point. We don't proceed in that case. So we, in our group, I have to admit that we have never worried about defining the mode volume very precisely. Because we're not really at a point, we've never really been at a point where we could worry about that so much. Eventually, he's of course absolutely right. There are subtleties in terms of defining the mode volume when you have a plasmonic type of resonator. And there are methods that people have developed, but we haven't really worried about it so much because, again, the level of experimentation hasn't gotten us to the point to worry about the details of that.
Great. A question from Joseph Asarelli. Is the resolution high enough, either at room temperature or colder, to measure protein deformation? For example, a ligand binding to a receptor.
That's a very good point. So I imagine that the question refers to the final part where we're doing this type of experiment. So the resolution that you have here in terms of mass accuracy, let's say if we detect something of the order of 50 kilodalton, then you could have perhaps 10–20% mass resolution. That is not very difficult to get. And with that, if anything else that adds to it or docks onto the protein can be detected. So if it's a single peptide, then I think we can't do that right now. But if it's something that is a few kilodalton or a few tens of kilodaltons, we can do that. Right. So I hope that answered that question.
Great, thank you. Can you comment on the reproducibility of single molecule coupling to your cavity structures in terms of fabrication or more scalable?
Right. So the way right now, this would be a good thing to use for the discussion. So this is a typical kind of spectrum that we get right now if we send light through a waveguide and detecting on the other side. So you see that we're coupling to a dozen of molecules, and you see that the height of these molecules, the dips are slightly different. And this has to do with the fact that each molecule is sitting slightly at different distance from the waveguide. So at this level, we do not have any control over that. We are, and other people are working on ways to actually place molecules at well-defined places. As we were discussing, Farnaz, this, of course, is interesting also in other areas of nanotechnology. But in our case right now, we have the fortunate situation that we can have many, many molecules, and we can go and choose the ones that we like. So here we would be working with this guy and with this guy, and not with this guy.
Great. So if there are no more questions, let me just double check. Could you, so a question from Vladimir Bolovich, could you describe the observation of strong coupling in your molecular structures?
Correct, sorry.
Describe the observation of strong coupling in your molecular structures.
Right, so the observation of, again, I don't have the data right now because we're just writing that up. But the observation, what you get is a clear Rabi splitting, and that also manifests itself very clearly in the nonlinear behavior of the interaction. So we're at a point where we can switch the system with one single photon. So reaching that single photon switching sensitivity is also a clear kind of a result of having entered a strong coupling regime.
Wonderful. So thank you, Vahid, for your time, for the wonderful talk. Thank you all for listening. There is one question asking about the availability of the PowerPoint slides. I just wanted to remind everyone that the MIT-nano seminars are recorded. So within a few weeks, they're going to be posted on the website. So you can rewatch them through accessing the website. Thank you, Vahid.
Well, thank you very much for having me.
Thanks.